now moving along to the genetic portion on Apollinaria spinifera. We wanted to determine if softshell turtles in northeast Alabama were genetically similar to a specimen collected by Wiesrock and Jansen in Macon County, Alabama in 2000. Since softshell turtles are prone to desiccation, their dispersal may be limited or restricted to adjacent bodies of water. So I'm sure some of you guys have never seen a softshell turtle, besides the other picture that I just showed. So I wanted to show you this little guy again. This is a male, because he's very tiny. And it looks like a chunk here. They're prone to bites from other turtles. Since it is on a golf course, possibly a weed eater, lawnmower kind of thing, whatever. But their leathery skin, you can see how it is different. They have a different thing going on with them. So this does make them very prone to drying out. So after collecting softshell turtles in the field, the turtles were brought back to the campus for blood extraction. Blood was extracted from the caudal vein or from a variety of tiny veins on the plastron of the turtle. When Dr. Murdoch wasn't available to get it from the sweet spot, the caudal vein, I had to sit there with the little needle and pinprick the tiny vein and then suck the blood. Pinprick, suck up the blood. So I learned from this I can never be a drug addict because I can't find veins, obviously. So it's good, it's very good. Samples were then centrifuged in 1.5 milliliter tubes, and the supernatant was discarded. DNA was isolated using a Kayla microDNA extraction kit, and purified DNA was used in an initial PCR for cytochrome B under the thermal conditions listed right here. PCR was conducted in 25 microliter volumes containing 100 nanograms of DNA, 1.5 millimolar of magnesium chloride, 50 millimolar of potassium chloride, 200 nanomolar of DNTPs, and 8 millimolar primers. This also contained 1 unit of Taq polymerase, and Taq actually prevents priming at room temperature. So it's a hot start Taq, so it only works once you actually get it into the PCR machine with a heated element. So you're not just sitting there and, as you get everything together, it's doing its thing, which is good. We will also want to use the same primers that Wiesrock and Jansen used. They actually developed them in their lab in order to be able to overlay our data with theirs. And reaction conditions were the same as Wiesrock and Jansen. Wait, sorry, go back. Oh no. Sorry about that. How do we just go back to that? I'm sorry, guys. It's like a turtle strobe light. Oh, I did it again.
Hit up.
Hit up? Oh, okay.
There you go.
Sorry, I didn't know this. Okay, so I wanted to also mention over here, we have this business right here. Where did never mind. I'm losing my mind slowly but surely. PCR product were separated on a 1.5% agarose gel, and the target was then excised. And this is a picture of my gel over here with the target 800 base pair reagent. And this is when we had 3 turtles at the time, so we ran 3 samples at the same time. So you can see this band right here. I'm not very good steady with the laser, but it's right there. And after cutting the gel, the desired location, we suspended the fragment using a Kayla quick gel extraction kit. And if you note on this picture as well, we have multiple bands, and so did Wiesrock and Jansen. They also reporting having to excise the fragment from a gel that had multiple products. However, they say that theirs is about 800 base pairs. So you're like, are we going towards 700 or are we going up more in 800? They're very vague about their description there. That's okay. Okay. We then took our purified DNA from the gel fragment to run an additional PCR in order to amplify the DNA. PCR conditions were the same as listed previously in the initial DNA isolation. And unfortunately, only one of the 4 samples amplified after the initial DNA isolation. Cloning was performed on that sample according to the topo cloning reaction protocol. And this was then transformed into competent E. coli using one-shot top 10 chemically competent E. coli one-shot chemical transformation protocol. And our colonies were allowed to grow overnight. And the next day, this is actually a picture of my plate. So the E. coli did its job, worked wonders. And we used these selected colonies as template for PCR. This allowed for the E. coli to work its magic and make more template. PCR, as I described, is like a tube full of bunnies producing lots and lots of DNA very quickly. So PCR products were run again on the same gel used previously, a 1.5% agarose gel. And colonies that actually have the correct recombinant DNA are 4, 5, 6, 9, and 10. And they appear to have the correct amplification in the 800 base pair region. We actually chose 9 and 10 for sequencing because they were on the end of our numbered spectroptenamycin plate, so we could just easily use those. So in order to grow up more plasmids, cultures harvested from colonies 9 and 10 were added to 3 milliliters of LB broth and 15 microliters of spectroptenamycin. This mixture was placed on the incubator and allowed to grow overnight. In order to remove the E. coli, plasmids were purified using a Kayla prep spin mini prep kit, and the DNA was eluted using EB buffer. We also quantified the DNA using a Biomate 3 spectrophotometer, and the spectrophotometer was blanked using EB buffer, because I have found out that if you blank it with water when you use EB, it is not read-written. So our colony 9 had 90 nanograms per milliliter, and colony 10 had 60 nanograms per milliliter. So colonies 9 and 10 were sent to the University of Alabama's Hal and Elizabeth Center Genomic Core Facility, and the samples were analyzed using the sequencer pictured here, which is an ABI 3730 DNA analyzer. Samples were processed, and the nucleotide sequence were actually available online the next business day. So we thought that colony 9 and 10 would have an identical nucleotide sequence. However, colony 9 was 713 base pairs long, and colony 10 was a different 760 base pairs long, neither of which were our intended mitochondrial DNA, and were both genomic DNA. We are still in the process of sequencing additional colonies, and when we first initially tried, we tried to do it without the cloning. So we isolated the DNA and we went directly to sequencing. And because we had those multiple bands, the sequencer said, you want to put an A and a T and a G all in the same place? We don't know what to do. We're going to give you like an N. So that is why we had to further go in, hope that the plasmids were going to do their wonders and, give us clear bands. But unfortunately, they did not sequence mitochondrial DNA. So we know that if we do continue this, that we will eventually screen a colony that has the correct recombinant information in it, and we will have our mitochondrial DNA that will hopefully match up with Wiesrock and Jansen's turtle from Macon County, Alabama.